Hello, this video will provide an introductory look at ribosome binding sites, or RBS. This video will hopefully provide you with enough information to effectively navigate this website and learn as much as you can about these amazing yet fundamental parts of genetics. In order to understand the role these sites play in genetics, you must first understand the central dogma of genetics. If you are viewing this video, we assume you already are intimately familiar with this concept. However, for the sake of redundancy, let's review. Okay. So the starting point for protein synthesis is DNA. DNA, or deoxyribonucleic acid, is a two-stranded molecule made of nucleotides arranged in a double helix. An enzyme called RNA polymerase makes mRNA from DNA in a process known as transcription. mRNA, or messenger RNA, is a single-stranded nucleotide molecule that's found all throughout the cell. This is the transition molecule in this fun process of making proteins from DNA. Once the mRNA is made, a complex molecule made of RNA and proteins called a ribosome binds to it. The ribosome is made up of two subunits, the larger 50S subunit on top and the smaller 30S subunit on the bottom. The ribosome links amino acids together as encoded by the messenger RNA, forming the protein. This step is called translation. The focus of this video, the RBS, is a specific sequence on the messenger RNA molecule, which our ribosome binds to in order to initiate translation. Without this site, the ribosome would never find the correct site on mRNA, and the protein would never be formed, rendering the DNA useless. Ribosomes, and therefore ribosomal binding sites, are found in every living organism, prokaryotic and eukaryotic. Furthermore, prokaryotic and eukaryotic organisms differ in their versions of ribosomal binding sites, which make them unique. Prokaryotes utilize the Shine-Dogano sequence, and the eukaryotes utilize the cap structure. The Shine-Dogano sequence is integrated within the messenger RNA, while the cap structure is added to the mRNA after the step of transcription in eukaryotes. In order for translation to take place efficiently in bacteria cells, a start codon as well as an RBS are needed. The start codon is a 3-nucleotide sequence made in the order of adenine, uracil, and guanine. The ribosome is made of two main portions as mentioned before. The smaller subunit, the 30S, on the bottom, and the larger subunit, the 50S, on the top. In translation, the smaller subunit binds to the mRNA on the start codon. Unfortunately, this system is not perfect, so the smaller subunit does not always find the start codon. That's where our Shine-Dogano sequence comes in. The Shine-Dogano sequence is a ribosome binding site that is made up of nucleotides on the mRNA. This nucleotide is rich in purines and promotes the binding of the ribosome. This portion lies 6 to 7 nucleotides down from the start codon and increases the likelihood that the small subunit will bind at the correct location. When the smaller subunit binds, the larger subunit finds its counterpart and begins making the protein we need. Next, we are going to explore the eukaryotic ribosome binding sites. Unlike their prokaryotic counterparts, eukaryotic organisms add their RBS after the mRNA is transcribed. After the messenger RNA is made, a complex known as a cap is added to the 5' end of the strand. This complex works in a similar fashion to that of the Shine-Dogano sequence, and then it helps the smaller subunit bind to the correct location on the mRNA. The ribosome binds to the cap on the mRNA and begins scanning until it finds the start codon. Again, since nature is never perfect, sometimes the ribosome scans too quickly and may miss the intended start codon. This is where eukaryotes dive a little deeper. To combat this mistake, a sequence called a Kozak sequence is found near the start codon. This sequence slows down the ribosome and helps ensure correct translation. Even though the Kozak sequence is technically not a ribosome binding site, it's super important in the process, and it has a cool name. Thank you for viewing this video. Please use the sites to navigate the website and explore different aspects of the ribosomal binding sites. If this video is awesome and you feel confident, go ahead and visit the educational resources link to take a quiz.